This is Unit 1, Chapter 2, the second part on experimentation in psychology. So just to review, remember when we look at experiments, the purpose of it is to isolate cause and effect. So determine causation when you have different variables. And so one of the things you want to do is control the factors. And you want to manipulate the factors that you are trying to study. And you want to hold constant or control the other factors so that they don't influence the results of your study. And one of the ways that you can help control some of these factors is through random assignment. Random assignment helps to hold constant all other factors, unlike in a correlational study, which can uncover naturally occurring relationships. An experiment manipulates a factor to determine its effect. There are two types of random assignment. There is blind, which is uninformed, and there are inside blind experiments. There are the single blind procedure and the double blind procedure. In a single blind procedure, participants are randomly assigned to research groups and do not know whether or not they're getting any treatment or whether they're in the control group. In a double-blind procedure, neither the participants nor the researchers know who is assigned to which group. And this particular type of procedure really helps eliminate bias. And it can really help prevent, for example, bias on the part of the researcher. So let's say a researcher is doing a drug study and they give a drug to one group and they know that that participant is in the group that's receiving the actual drug. They might treat that participant differently or look for different results that confirm the effects of the drug that they're hoping to see than say they would if they knew the person they were talking to and working with was getting the placebo. The placebo effect is where people believe a treatment will work simply because their care provider tells them it will. And it's really interesting about the placebo effect because what they have found is the more expensive the perceived placebo is, or the drug per se, the more likely people believe that it will work, the stronger the effect. So if they believe that they are getting the drug, and when in fact they're getting the placebo, and if they believe that that drug is actually very expensive, they're more likely to think that it is the drug and that it will work. So the placebo effect is actually stronger. And of course, again, when we're talking about random assignment, you have two groups. You have the experimental group, which receives the treatment, or the independent variable in a study, and the control group, which does not receive the treatment. So let's talk about the different aspects of an experiment. You have an independent variable, you have confounding variables, and then you have the dependent variable. So the independent variable is the manipulative variable because it can vary independently of the other factors. This could be something like age or weight. The confounding variable are other factors which can potentially influence the results of an experiment. And the dependent variable is whatever is being affected by the independent variable. So that's what's being measured. Both the independent variable and the dependent variable are given operational definitions which specify the procedures that manipulate the independent variable or measure the dependent variable. I'll explain this further in a minute. So in other words, they have operational definitions so they are easily measured and can be replicated. The dependent variable is the measured variable. And this is what the researchers expect will occur in the study. So whenever you're looking at an experiment, you look for that first. When you're identifying variables in a study, you always look for the dependent variable first. What is it they expect will occur? The independent variable is the manipulated variable. That's the thing that changes, and so you should look for it second. And I'm going to explain this in just a minute. Also, remember that confounding variables, these are things that can interfere with the assignment, with the experiment. So when you do random assignment, The purpose of that is to kind of control the effect of those different variables or those other factors which could interfere with the study. Okay, so let's look at this. All right, this is a study where they are trying to see if breast milk increases a child's intelligence. Okay, and so the operational definition for the experimental group is it's clearly defined that those children are going to be breastfed. The control group, on the other hand, is going to have formula. Okay, now they are going to use random assignment to help get rid of some confounding variables. For example, being in a good home environment, having parents that are highly intelligent. And how they're going to do that is they're going to randomly assign the children to either the experimental or the control group. And by doing that, they're hoping to evenly spread out children from these different backgrounds. Okay? Now, the independent variable is the second thing you're going to look at. The first thing you're going to look at is what are they trying to find out? And what they're trying to find out is the intelligence score of a child at age 8. Okay? That's what they're looking at and how it changes. What they are going to manipulate is the second thing, and that is whether or not they have the breast milk or whether they're formula fed. Okay? So the dependent variable is what they're at the end looking for, the result. The independent variable is what they're doing or changing in order to affect the result. Comparing research methods, we've talked about these different kinds. You have the 3 different types. You have a descriptive method, right? And that's your case studies, surveys, naturalistic observations. Those are just observing and recording behaviors. Remember, single cases can be misleading. They can't be generalized to the overall population. You can't control variables. So there's lots of issues there. Correlational studies look for naturally occurring relationships and to try to figure out how well one thing predicts another. However, they don't talk about causation at all. And so this is when you have a lot of data. So this is when you're looking for statistics, correlational coefficients, etc. And then an experimental research method, you're looking for cause and effect. So you're looking for causation. So you manipulate one or more factors, right? The independent variable. You use random assignment to get rid of confounding variables or other factors that could influence your study. The importance about this is that it specifies its cause and effect and the variables are controlled. Now, looking at statistical reasoning in everyday life, statistics. Here's an example. When we describe data, we talk about these are measures of central tendency. Okay? Measures of central tendency attempt to summarize data. They always look at the measure of central tendency being used in a study. So are they using mode, mean, or median? And consider whether a few atypical scores are distorting it. Okay, so here is an example. So here you have a bunch of scores, right? Income per family in thousands of dollars. In this case, the mode, that score, 40, occurs the most times. And the mean is the 140, right? The average. And the median is the middle score. Now, when they talk about atypical scores, they're talking about outlier scores. Is the mean being distorted because you have one really high score of 1,420? Okay, so you want to look for that whenever you're looking at measures of central tendency. Are there a few scores that are skewing your data? You always want to be mindful of that. We also look at variability, and we do that with range and standard deviation. So the range is going to be the gap between the lowest and highest scores. Standard deviation tells us how much an individual score differs from the mean. So again, standard deviation tells us how much individual scores differ from the mean. The higher the standard deviation, the less similar the scores are. Test scores have a standard deviation of 5. In that case, everyone scores similarly. If the standard deviation is 50, the scores weren't similar at all. So the smaller the standard deviation, the more similar the scores are. The higher, the less similar. If you have a smaller standard deviation, you can draw more stable conclusions from the data. So the smaller the standard deviation, the more predictions and conclusions you can make on the data, or the more reliable probably the information is, the results of the study are. This is a bell curve, okay? So a normal bell curve. And this is how we use to measure variability. It's a pretty common form. You do this a lot with large numbers of data. And in this case, when you have a bell curve, the idea is that most cases will fall near the mean, and there will be very few outliers. So in a normal bell curve, 68% of the scores are within 1 standard deviation. 95% are within 2 standard deviation points. Another thing we talk about is making inferences. When is an observed difference reliable? So, of course, representative samples are better than biased samples. Generalizations based on a few unrepresentative cases are very unreliable. So you can't make generalizations on a few unrepresentative cases. So the less variable the observations are, the more reliable than those that are more variable, of course. When we look at statistical significance, the differences between the averages is relatively large. It does not imply the importance of the results. So a statistical significance refers to how two groups measure different. If you graph the data from two groups and the graphs didn't overlap or overlapped only a little, then the difference would be significant. Significance allows you to say how likely the difference in the means is due to chance. And the usual goal is to get a significance level of 0.05, which says the results are only 5% due to chance or 95% due to your independent variable. So you want a small score of statistical significance, so that way you can guarantee that it actually was your independent variable that got you your results. So basically what I just want you to know is statistical significance is that you're trying to get a level of about a 0.05 so that you can make sure that your results are not due to chance. Some questions we ask about psychology is, can laboratory experiments illuminate everyday life? What can they tell us about everyday life? The purpose is to test theoretical principles, and psychologists' concerns lie less with unique behaviors than with discovering general principles that help explain many behaviors. So they don't want to just look at an individual's causes for their behavior. They want to looking at big generalizations. What can they learn from this study that they can apply to a large population? Also, even when specific attitudes and behaviors vary by gender or across cultures, as they often do, the underlying processes, biology, are much the same. And that's important to remember. So culture and gender can affect behavior, but the processes that cause that behavior, the biological processes, are the same. The last piece I want to talk about is ethics in animal research. And we use animals to learn about people. Human physiology resembles that of many other animals. And this has led to treatments for human diseases such as insulin for diabetes. There are several issues, though.